Hi all, welcome back to Microbiology Lectures. Today we're just going to continue lecturing about growth rates of bacteria and archaea, for that matter, microorganisms, and some of the limiting factors that kind of limit either their growth or can optimize their growth. And the first one that I want to talk about is the temperature requirement that we see when we look at bacteria, or archaea for that matter, for microorganisms in general. And that is because often when we're talking about temperature, or the optimal temperature an organism can live at or grow at, we are interested in the relationship between temperature and their proteins, or temperature and their amino acid sequences. Because what happens under extreme temperatures, so as you're moving away from their optimal temperature towards higher temperatures, what happens is those amino acid chains will start to break apart or separate. And when they do that, it allows water to kind of seep in, and water will bind up to those amino acid chains, which basically changes the shape of the original protein. This is extremely important for things like proteins, which are ports or transmembrane proteins that go through the membrane, or enzymes that might have different allosteric or different sites that are very specific to a single molecule. You change the shape of it, and those proteins no longer work. And that's what we call denaturalization or denaturing of a protein. And so it's important to note that this amino acid sequence is determined by the genes or the genetics of that microorganism. And when we're talking about bacteria and archaea, their plasmids can have a direct effect on this amino acid sequence. So if the bacteria is now subjected to extreme temperatures or hotter temperatures than normal, and the plasmids can code for a protein or a series of proteins that can withstand those temperatures, then that organism has a better chance of surviving. And so as we go, we'll kind of talk about some examples of what it means and the importance of looking at temperature in regards to bacterial growth. So one big example, and one that the food services look at and examine all the time, is food preservation or prevention of spoilage. Okay, and so when we're often designing or talking about food-related illnesses, or how companies should store their food or prevent spoilage and things like that, we're doing it from a perspective of, well, what can grow at those different temperatures? What causes the food to spoil? And what's the temperature at which they can grow? And so we know that actually 4 degrees Celsius is a really good temperature, because it's above freezing. So you want to refrigerate the material. You don't want to freeze it. Freezing will prevent microbes from growing. It doesn't kill the microbes, but it prevents them from growing. But most of the microbes. But a temperature of around 4 degrees Celsius keeps the mesophils down and doesn't allow them to grow. And even the psychotrophs, it still keeps them from growing really fast. Okay, and we'll look at these guys in a second. The other thing that we might be interested in is from a disease perspective. We know that different bacteria, even on a human's body, because our body has different temperatures, like your extremities, ears, nose, toes, fingers, they're all cooler, or typically cooler, than things like the crevices of your body, like your groin region or your armpits or region like the inside of your mouth, these kind of things. These are all warmer than the extremities. And so we know that there are certain bacteria that grow outside on the extremities, like Microbiome leprae, which causes leprosy. And that can be a bacterial infection in the ears, hands, feet, fingertips, that kind of thing. They do best on cool environments, and then Hinderdentis supervative, or HS, requires warmer environments. So like you get these bacteria-filled pockets that occur in the armpit region, the groin, the buttocks, that kind of stuff. And so from a medical perspective, it's kind of important to know the temperature requirements. And so when you're documenting, you're trying to find out, well, what kind of infection is it? What might be the species that is causing this? Knowing something about where that species or that optimal growth of that species is can determine what the species might be that's affecting the individual. So, and that brings me to basically a species-specific kind of temperature range. So each species of microbe will have their own temperature range. Often we can truncate these into categories, and that's what I'm going to present here, are the different categories of temperature ranges. And so things like the psychrophilies or the cryophils, they have a low temperature range, so they can tolerate freezing fairly easy and still grow under freezing conditions. But for the most part, if you're going to find them in native habitats, you got to go to cold environments. And you can see that we typically break this up into 5 or so different groups. And so you have these psychrophilies down here, and then you might have the psychotrophs, which are the ones that we're interested with when it comes to food spoilage. And so we know that if we can keep our temperatures below 15 degrees Celsius, if we can keep our temperatures below room temperature, then the optimal temperature for the psychotrophs is the optimal growth is less. So their optimal growth is right around room temperature, right around that 22, 24 degrees Celsius. And if we can keep like our refrigeration at 4, then we can control their growth rate. It doesn't mean that they're not growing. They are, and that's why food does still spoil. But they just grow at a slower rate in like refrigeration. Along the same lines is the mesophilies, or the middle temperatures. A lot of the pathogens that we see for especially humans fall into this category. Okay, and so pathogenic bacteria, things that cause diseases or infections, those kind of things, fall basically between 35 and 40 degrees Celsius. Remember, humans maintain a body temperature right around 37, so that's like the sweet spot. So 37 is like the sweet spot for pathogens. And so inducing a fever can get them out of that sweet spot and cause some of them to start denaturing their proteins and cause a little bit of relief from a bacterial infection. We'll talk about kind of the innate immune system and development of fevers and things like that as a control mechanism for bacterial infection later. But that's a potential control mechanism. But just knowing that they have these temperature requirements means that the pathogens often they can't survive at even room temperature. So if it's on the countertop or something like that, it's not going to be affecting anyone just because they can't survive. The thermophils, they're important because they have very unique properties along with the hyperthermophils. So both these groups are typically found in extreme environments, hot springs, thermal vents, these kind of things. And lots of individuals study these constantly, like whether we're talking about like Yellowstone hot pools and how they get these very unique colors, a lot of which is caused by these hyperthermal fills or even thermal fills. Again, when we first started describing archaea, we stuck pretty much all archaea into the hyperthermal fills, but realize now that I don't want to call it a mistake, but we now know that there are archaea in all these different categories, and that they're not just the hyperthermal fills. But that being said, a lot of archaea occur in this because remember, we go back to that monolipid layer that some archaea form, where they have two phosphate heads and their fatty acid tails are actually linked and tied together. That helps them prevent kind of movement under hot conditions, and it helps them stabilize that lipid layer from being pulled apart under hot conditions. Another requirement, or another way at which something might play an important role in the growth rate of bacteria, is oxygen requirements. And so we talked about this a little bit before, when we are interested in growing organisms that need oxygen, especially in a closed system. We talked about the closed system where no new nutrients are coming in, no waste products are leaving. It may be important. It depends on how you want to grow the organism or what you're looking at. It may be important for that vial or that material to be shaked or agitated, so you can increase the amount of oxygen that's being mixed in the solution. Otherwise, what will happen when you don't do that is you'll get the top of the tube will go aerobic, which means there's oxygen transfer from the atmosphere at the top of the tube and the first layer of your, in this case, most of the time, liquid broth. And the bottom of the tube would go anaerobic, and there's no oxygen. And we know this is important. This relationship is important for things like biofilms, because you can get biofilms that occur in natural systems where the top portion of the biofilm that might be interacting with water or the atmosphere is aerobic, and the inside portion of the biofilm, which might be hidden away from the atmosphere or the water, is actually anaerobic. And so you get this kind of layering of the system, and you get the same thing in a test tube. So the position will often indicate oxygen requirement. And so sometimes this is an important test to find out whether we're dealing with aerobic or aerobes, or we're dealing with anaerobes. And so often we stick the term obligate to mean that they have to have. So in this case, it's an obligate aerobe if it has to have oxygen. You might find that sometimes people just drop the obligate and they just use aerobe, that term. Okay? If it's an obligate anaerobe, it means it can't live with oxygen, so no oxygen. And in fact, often an obligate anaerobe, if oxygen is present, the oxygen is toxic. So it messes with the way at which they generate energy. So remember that oxygen, the reason it's present, the reason it's needed for most of these organisms, it's a vital gas for respiration or for the process of generating energy. And a lot of times it's the electron acceptor that picks up those electrons and hydrogen ions, and it's bound together and it generates metabolic water. And so it's really important, but in systems where they might be using a different way, a different process to generate energy, oxygen can sometimes be gas that when it gets in, it causes problems with the system, and it might bind to something, and it doesn't allow the system to work any longer. Okay, and there's lots of ways at which we can just label other patterns that we see, like a facultative aerobe or a facultative anaerobe would mean that they can use oxygen but they don't require it, or a facultative anaerobe might be they grow better without oxygen but they utilize oxygen occasionally. So it really just depends. And then there's other ways that we can classify these organisms as microaerophils, meaning they can use a small amount, or aerotolerant anaerobes, means they can tolerate oxygen because oxygen doesn't get in, it doesn't diffuse into like the cytoplasm, and it doesn't haywire their system. So lots of ways that we can look at this. This is just another way to kind of classify them. Normally we kind of classify them in 5 classes, although there are situations where they don't fall in these 5 classes. But again, an aerobic bacteria is going to use oxygen for their energy process. A facultative anaerobe can use oxygen, okay, but it doesn't have to use oxygen. So it's both, okay. An obligate anaerobe will never use oxygen for its energetic process. It can't grow in the presence of oxygen. So in the case of an obligate anaerobe, it means that oxygen indeed is lethal to them. Okay, and then a microaerophil would mean that it needs just a little bit of oxygen. And so maybe a small portion of how they generate energy requires oxygen, but not all of it. So they might do a lot of fermentation processes that don't need the oxygen. Okay, and then an aerotolerant anaerobe would just mean they tolerate oxygen, but it doesn't use oxygen. Okay? So let's look at what that might mean and why that's important. Well, there are certain tests that we can use instead of their presence or where they're at in a test tube. We can look at the production of certain chemicals. So when we're talking about aerobic respiration, remember they're utilizing oxygen for the purpose of it helps with the generation of energy. Well, in the process of bringing in oxygen and utilizing oxygen, often superoxides are generated. Right? And so these can be the result of taking in oxygen, but also cleaving things like carbohydrates, which would have oxygen, breaking them apart. A lot of times you'll get these superoxides that are produced. And superoxides are they're harmful. They're a lethal product. They cause these oxidative products will cause damage to cell membranes and to protein compartments and things like that. Along with that is the relationship between superoxides and hydrogen peroxides. A lot of times these superoxides, because they're toxic, they'll be converted, and their first conversion is into hydrogen peroxides, which are also toxic. And so they can damage cellular components. They can bind to things, so the active sites don't work anymore. They can change the shape of proteins. There's lots of things that these byproducts can do. So, in order to deal with them, most bacteria that utilize oxygen in some form of respiration and for energy production, they have specialized enzymes. So they have genetic codes for specialized enzymes, one being the superoxide dismutase, or SOD, and that superoxide dismutase will convert radical oxygens or superoxides into hydrogen peroxide. And some water and oxygen, etc. Okay? But that hydrogen peroxide, again, that's a toxic chemical also. So not only do they often produce superoxide dismutase, they also have to produce catalase. And these catalase will take these hydrogen peroxide molecules and break them into water and oxygen. So when we're talking about aerobic bacteria, instead of doing a test on where they are in a test tube, which might not separate aerotolerant anaerobes from aerobic bacteria, one way to get at that is you can just test for the presence of catalase or the presence of sod, and so that can give you it can be a useful test to kind of separate anaerobes that are tolerant of oxygen and aerobic bacteria that are utilizing oxygen. pH requirements. Okay, so a couple things with pH. Of course, bacteria themselves survive in lots of different pHs. Some of this is self-generated and can change because, remember, bacteria are utilizing proton pumps and proton motive forces to do things like move their flagellum to generate ATP synthase. And so these protons, they can be pumping them across and changing the environment right around the cell, but also they live in places where they're exposed to a certain number of protons or a certain amount of acidity or alkalinity. Most microbes, fairly easy to tell that they're neutrophils, so they occur with an optimum around a pH of 7 or neutral water. And so we've been taking advantage of this for a very long time. We can preserve things in acids or for bases for that matter if you want. But most of the time we use acid to preserve things. And that will not allow a lot of the microbes that might cause food spoilage to take set and actually grow if we preserve things in acidity or an acid solution. Okay? Now there are some, like pylori, which often is the cause of ulcers, stomach ulcers, that do really well in acidic environments. And they can be very important. Pylori can help break down large carbohydrates, but also tends to help with the production or decrease of ammonia-based products. And so they can be important for the body, but they can also cause ulcers. But they live in regions where they'd be considered acidophils or acid-loving bacteria. Okay, so you have these acidophil that basically between 5 pH and 1, although there are species of bacteria that live in environments that are less than 1. Okay, neutrophils right around that 7, and then that alkali fill would be an optimum around 9.5, somewhere in there. Okay, so acidophils, most of the time, like I said, there are some special cases where they're growing less than 1. Okay, so Picrophilus ashime was a bacteria that was discovered in Japan and some hot springs there, and these pools have a pH, a very, very acidic pH, and sure enough, there's bacteria that live there. Okay, but there's alkali phils that normally they have a pH above 8. These can be very important for pharmaceuticals, but also very important for industry. We utilize a lot of the enzymes from these alkalophils to produce things like detergents, and different enzymes in our detergents which helps break down stains and whatnot, and so they seem to be fairly important for that. Okay. Water availability, huge one. Okay, really important. We've known about this for thousands of years, that bacteria, archaea, microbes, microorganisms in general require water, like all organisms on this planet require water. And so we can take advantage of that by using things like salt and sugar to increase the solutes around the outside of a piece of food, so maybe a meat or something like that. We think it was probably discovered in Egyptian time, where they would catch a fish and maybe they would rinse it or carry it in salt water, and the food that was rinsed or had been carried in salt water tend to last longer than the organisms that were carried in fresh water. And so they put two and two together, and they started saying, all right, well, let's dump salt on it, and that seemed to preserve the food. And then they utilized sugar also, and that was just it just changes the osmotic relationship. So it causes all the bacteria that are housed on that food to lose the water inside their cells and dry up. Now, in many cases, this is enough to cause the bacteria, if not to die, to be destroyed, at least it prevents them from growing. So you might have a limited number of bacteria that's on a piece of meat, salt preserve it, and now that bacteria is still there, but it's not to the numbers that would ever inoculate you or cause illness in you if you were to eat it. We do know that sometimes adding salt to certain bacteria will cause them to go into an endospore status, but that's just a limited number of species that we know about that do that. But even at that, it's probably still worth the effort to preserve with salt, because it made people not get sick. Okay. Now, there are some microbes that really enjoy high levels of salt, or they can withstand it, or in some even they live in it and thrive in it. Okay, so like Staphylococcus on dry, salty skin. There's quite a few species that can withstand even to above 10%, but most marine species are withstanding things in the 3.5%. The extreme halophils, like of the Dead Sea, Utah salt flats, those kind of things, they might be in environments where 35% salinity. So one thing is if in a lot of these environments, if they're going to withstand it and they're going to survive, then they need to pump. If it's a salty environment, they need to pump salt in, and they need to store salt inside their cytoplasm. Okay, and if the more salt they can store inside their cytoplasm, if they can get a higher concentration, say than 3%, okay, so if they get a higher concentration here, then water will flow in, and they can just keep storing salt and utilize that process. If they're in an environment and the inside is got less solutes than the outside, and then of course water will flow out. So a lot of these organisms, if they're going to live in these extreme environments, they're going to store huge amounts of solutes inside to prevent desiccation. Okay? And so here you can just see a picture of the Dead Sea and all this along the bank. This is all salt, but we found lots of species of bacteria that can live in these extreme environments, and that's what they're doing. They're withstanding osmosis by storing salt within inside their cytoplasm for the most part. Okay. The next piece that I really want to talk about is nutrition, and we will talk about that next time.